Good morning, everybody. Who do we have here today? Oh, the usual suspects. All right, so happy Friday, everybody. Woohoo! Yay! Happy dance. And it's Halloween this weekend too, so I'll get to steal lots of my kids' candy, which is pretty awesome. Easter's also pretty good too. Although they're now in the habit of counting their stuff, which is kind of unfortunate. Never mind. All right, so this is kind of where we ended up. And I do keep meaning to replace this kind of chicken scratching picture of a transcript, but it actually does just fine, right? Pictorially. And so essentially, if we go back to, and I like bouncing around a bit because I like tying things together. All right, so this is essentially what the gene would look like. This is what the pre-mRNA or the nascent mRNA would look like, right, with its introns and the like. And once it's processed, it'll look like this, right? So every eukaryotic mRNA that gets exported to the cytoplasm looks something like this. 5' cap, 5' UTR, untranslated region. Open reading frame or coding sequence, which is bookended by our start codon and a stop codon, right? And it's one of these. So there are 3 possible stop codons. You know, one of them is going to be there because you have to stop translation somehow. And also, just as a kind of a side, you know, these stop codons and start codons also apply to prokaryotes, right? The difference in prokaryotes is that they don't have all these other structures. They're actually kind of quite a bit weirder, to be honest, in some ways. Then downstream of this coding sequence, which is what's going to get translated into protein, because it's coding for protein, are the 3' UTR and the poly A tail. A little bit of revision for you. What's the 5' UTR important in? And they're actually 5' UTR and 5' cap. So what's the purpose of those 2 RNA structures? Coffee sipping time. What's the purpose of 5' cap and 5' UTR? No, it doesn't stop transcription. Good guess though. That's okay. That's why we're doing revision, right? Remember the whole find out what you don't know before the exam rather than in the exam is the key. Okay, so both ends of the transcript must be what from RNases? Made available to RNases? Protected from RNases? What do we need to do to the, why do we have a 5' cap and a poly A tail? Yeah, they've got to be protected. What else does the 5' cap and 5' UTR get involved in? So protection is one aspect. Protection is stability. What's the other? What's the whole purpose of producing an mRNA? mRNA doesn't do anything in and of itself. What's the next step? once that transcript has been exported to the cytoplasm, what happens to it next? Right. Yeah, it gets translated into proteins. It doesn't get made into proteins. You have to be a little careful on language. Right, we don't turn the RNA into protein. We use it to make protein, like an instruction sheet. Right. So the 5' cap is very important in the initiation of translation. The 3' UTR and poly-A tail, pardon me, are also involved in the control of translation, right? So they have multiple functions. It's not just about stability and protecting the ends, it's also about the next step. Okay, so speaking of the next step, see like the nice segue there. Translation, right? So both eukaryotic and prokaryotic mRNAs get translated, obviously. There are some differences. There's a lot of commonality as well, right? And so essentially to make protein, you need 4 different things, right? You obviously need the mRNA, right? Because that's the information, that's the instruction set on how to make that protein, right? You need ribosomes, right? So these are another example of riboproteins. What's the other riboprotein we've come across so far? Or proteins? Actually, there's a whole bunch of them.
Like you. I'm glad it didn't work with her because she was not confident. She was just very like dependent on whatever I told her.
You
It's
might
like, "You're
need
not
to
like
mute
that."
your microphone, whoever's chatting.
Like, yeah, well, I've been single since 2018, so I can't really depend on anybody telling me anything.
Gina, you might want to mute your mic. Awesome. Armando, you're even faster than I am. Yeah, that's all right. I don't mind. No worries on my part. Anyway, so back to the question. What's the other riboprotein? We've seen so far. Hint. Bunch of them. And here's an even better one. I made a bad joke about Irish, oops, if I can spell it right, rock stars. Come on, that's got to be a giveaway. Also pronounced. Hey, yeah, there you go. What's their collective name? What's the collective name for U1, U2, U4, 5, and 6, I think. I think there's 3 that's missing. Right, they form the spliceosome. Ah, here's another hint. Yeah, SNRPs. That's it. Can anybody remember what SNRP stands for? Let's see. Shall I type it out? A little bit of self-punishment going on here. They are, hang on, get this up. Small nuclear ribonucleo. Oops. Oh yeah, thank you, Armando. You saved me from that. That takes a long time to type. Exactly, they're small nuclear ribonucleoprotein particles, right? So those are, thank you. Those are also a protein RNA complex, right? So ribosomes are another example of that. There's also the tRNAs, right? So these are called transfer RNAs, and I actually always feel like they should be called translator RNAs, right? tRNAs are what translate the nucleic acid information into amino acid sequence, right? So these are very much, in my mind at least, the equivalent of the Rosetta Stone. Anybody heard of the Rosetta Stone? It's actually a brand of language software, language learning software too, for this reason. Has anybody heard of the Rosetta Stone? It was a major find found in, or somewhere along. Yeah, so it's the tablet. That's right, Isaiah. It was a tablet which allowed Egyptologists to decipher hieroglyphics, which were completely impenetrable, understandably so. And they found this stone tablet basically had a whole bunch of hieroglyphs on it, and also their meaning in, I think, like Sumerian or something like that, one of the dialects at the time, the local languages. And so that allowed them to translate hieroglyphs into modern languages, right? And so the tRNAs essentially do the same. They are the key to this whole process. They're actually what makes it work, right? And they do so because they have, where do we have a tRNA? There we go. tRNAs are obviously made out of RNA, right? So they can base pair with the mRNA, right? By base pair complementarity. But they also, and they do that through this bit down here called the anticodon. But they also carry on their other end an amino acid that's very specific to that anticodon, right? So this is like the intermediate. This is the link between nucleic acid and protein. Right, they're really what makes all work. And then the thing that allows this to be such a specific process are this kind of mouthful of letters, which are amino acyl tRNA synthetases. And essentially what they do is they stick the correct amino acid onto its tRNA. We'll get through why that's so important and how that happens in a little bit. Okay, so these are the kind of the central components. And again, the aim is to turn the information in mRNA into a series of amino acids called a polypeptide, which then forms a 3D structure that does something, right? And so we go from relatively simple linear molecule of 4 bases, right? A, U, C's, and G's. And we end up with a complex 3-dimensional structure, right, which can move and get changed and go places, that consists of 20 different amino acids, sometimes thousands of them, right? And there are tens of thousands, around 20,000, give or take, genes, protein coding genes in our genome, right? All of those genes, all of those proteins are encoded by mRNA and then DNA with 4 bits of information. So a big part of the complexity of what I'm going to be teaching now is how we go from 4 bits of information to 20 amino acids, and I'm not going to try doing like 20,000, to 20,000 different proteins, which all have different structures, different properties. They go to different places in the cell. They do different things. They interact with other proteins, form complexes like ones we've seen already in terms of, you know, DNA and RNA polymerases, right? And so it's really quite astonishing to go from that degree of simplicity to that degree of complexity. And translation is what sits bang in the middle of that. So it's a, you know, a very complex process as a result. Okay, so first up, we need to go through what an actual protein is, right? I've actually found a neat and natty video, which I'll add to this in a wee bit. And so basically proteins are, they're linear, well, let's see if I can find a picture rather than just using bloody words. Oh, if I can spell. What do I want? I want day in life of a motor protein. Funny the things that come up in my Google auto suggest. That's actually, if you're a molecular biology nerd like me, it's actually a pretty cool film. You can go watch it sometime. Primary, secondary, and tertiary. Let's see if that brings up what I'm after. Yeah, good enough. Let's find a nice picture. No, that doesn't do what I want. I want do. There you go. This will do. Dang it, where's that nice picture gone? Oh, this is like being on dial-up. Nope. All right, anyway. So essentially a polypeptide is this, right? It's a linear string of amino acids, right? Which amino acid is in what place is dictated by the mRNA. So each, depending on the protein, which obviously depends on the mRNA and the gene that it's transcribed from, you get a different sequence of different amino acids, right? But the primary structure is this linear polypeptide, right? So this is not functional yet. It then gets folded and twisted, right? Depending on the sequence of those amino acids, like what you have in what order. And then those structures, which are called alpha helices and beta pleated sheets, then get folded again to create the tertiary structure, right? Which is what we have, where be that? Here, right? And so this is now a three-dimensional structure, right? It may also be form a complex with other proteins. So let's see if I can find my fave quaternary structure. I wonder if I can get a GIF. Oh no, I don't want that nonsense. There you go. Is this going to work? Oh, that's kind of fancy. So this is a model of hemoglobin right in the center there. And this is what's called a ribbon molecule of protein structure, right? So what you're seeing is kind of the polypeptide chains and secondary structures kind of expressed as ribbons, right? There are also things called space filling models, which look like this, right? So you're actually modeling the space which is occupied by those different things. But anyway, oh, that's actually kind of neat. This is a complex of 4 different proteins: alpha-1, alpha-2, beta-2, and beta-1 hemoglobin, right? So those are encoded by 4 different genes. They are all transcribed, translated into this linear polypeptide that's folded into this beta pleated sheet or alpha helix or loops. And then those are folded into their own kind of 3-dimensional structure. And then those 4 different proteins are stuck together in a much larger structure, which actually does something, right? So these individual proteins on their own don't work. They have to be complex together to do their job. And what this is actually shown, which is quite why I was looking for it, is that the hemoglobin protein that's in your blood cells, in your red blood cells, that's kind of why oxygenated blood is red. They have a heme group. Actually, they have 1, or is it 4? Can't remember. I think it's 4 heme groups. But either way, they have them right at the center of this molecule. You can kind of see it better over here, right? And that binds oxygen, right? But it only binds it in a certain conformational state. So you can see that this form here, right, is going to blue, from red to blue. It says oxy and then deoxy. As the protein changes shape, it changes its affinity for oxygen. So in one particular shape, it binds oxygen. In a different shape, it releases it, right? And that's essentially how your breathing and your metabolizing and, you know, your consumption of oxygen and production of CO2, how that's balanced by hemoglobin, right? Which takes that oxygen from your lungs, from the air essentially, and then carries it to your metabolically active tissues like your muscles, and then dumps that oxygen there, which is then used by those cells. So hemoglobin, super cool. Fave protein. Also, fun fact on a completely different tangent, the heme group in hemoglobin is quite toxic, right? So malaria parasites, which infect red blood cells, they feast on hemoglobin because it's yummy and there's a lot of it. But they have to put that heme group into a separate kind of organelle, and that's called the what's it, apicoplast. And it's very similar to chloroplast mitochondria, but it's this kind of distinct organelle, and they put it there, and then it doesn't cause them any harm. It's like a kind of safe space to put their hemoglobin in, or their heme. Anyway, that's just another wild and wacky tangent. So this is the end product, or if it's a more of a complex multi-unit protein, then this is the kind of penultimate step. This will then get added to something else. But we start with amino acids, right? And so essentially, this is a string, right? And so we're not showing the individual amino acids here, we're showing the secondary structures. It's a string of individual amino acids, right? Which, you know, kind of coil or pleat themselves, or, you know, kind of wave around in the breeze, depending on their individual characteristics, right? And so amino acids are essentially they're the building blocks. They're like nucleotides, right? And much like nucleotides and nucleic acid, they have part of their structure which is common to all of them, like the phosphate ribose or the phosphate sugar part of nucleic acids, and part which is unique to that particular amino acid. So as you have A, T, C's, and G's in DNA, right, that's the nitrogenous base which hangs off of the first prime carbon amino acids have an amino group, an alpha carbon. It's called the alpha carbon, not because it's like dominates the rest. It's because it's the one that's in the center. Everything hangs off of it. And a carboxyl group. And so these actually give us direction and a bit of terminology, right? So let's see if this will auto correct me again. No, it doesn't. Good. So the amino group is always at the start of the polypeptide. And that is called the N terminus, right? From the N in amino. And then, well, the nitrogen is another way of thinking about it too. The carboxyl group equals the end of the polypeptide. And where new amino acids are added, right? So this is the equivalent of the 3' OH of a nucleic acid. And that's called the C terminus. Right, so amino acids and the polypeptides that they form, essentially, have direction, just the same as DNA and RNA do. Right. So, and as the same as DNA and RNA, these amino groups, carbons and carboxyl groups form the backbone of the protein, right? So just as you have phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar in DNA or RNA, sorry, I got something stuck in my finger. You also have in polypeptides, NCC, 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 like all the way along, right? The thing that differs is the R group. Now, some of you may already be preemptively rolling your eyes at this, but because it's Friday, I'm allowed. What is a pirate's favorite letter? What's a pirate's favorite letter? Dad joke time. No, it's the C. That always makes me laugh, even if it doesn't make anybody else laugh. Always makes me laugh. Get it? Because they live on on the sea, sailing, robbing other ships, stuff like that. Anyway, I think that's pretty funny. Oh, just because it's like two for the price of one deal today. My daughter told me this. When does a dad joke become a dad joke? This is like higher level dad joke status now. When does a dad joke become a dad joke? When it becomes apparent. How cool is that? Get it? Apparent. Yeah. Anyway, whew, tough crowd, you lot. So, all right, I'm gonna spare you from any more dad jokes. I thought that was a nice little kind of break halfway through the class. Pretty well timed. So, I'm gonna have to tell my wife about this. You realize that how you are totally unappreciative of my dad jokes. She'll give me some sympathy, I'm sure. So, essentially, amino acids are added together to make polypeptides, kind of in the same way that oh, good. Thank you, Melody. Oh, that's very sweet. That's made my day. Kind of in the same way as you add nucleotides onto a growing DNA or RNA chain. You have an OH at one end, right, on the end, literally, and you have a hydrogen on the incoming molecule, amino acid in this case. Those are removed to create water, and then in the process, you form a peptide bond, right? So basically, peptide bonds are to proteins or polypeptides. I use the term polypeptide as like it's like a precursor thing. It's like pre-mRNA essentially, right? It's not the finished product, it's what's being produced by the ribosome essentially. So peptide bonds are to polypeptides. Hang on, sorry, I have to see what I'm writing here. What phosphodiester bonds are to RNA or DNA, right? So they are the bonds that hold the individual components together. And so one thing that's worth pointing out too, right? The bonds that hold these amino acids together are very strong, right? They're covalent bonds. The bonds that hold this structure together are not necessarily very strong, right? And that's how if you put too much heat or you kind of change the aqueous environment or pressure or whatever, right? You can actually force this protein structure to kind of change and fall to pieces. It's called denaturation. And that is essentially what you do when you fry an egg, right? So when you fry an egg, when you crack it into a pan, the egg white is translucent, right? Or even transparent, right? And it's that way because albumin, which is the main protein in egg white, has a particular structure. So it looks like something like this. Now, when you heat it, some oil, with your spatula, what happens is that heat causes those bonds that hold it together in that particular shape to break, and then it changes shape, and when it cools down, it sets in a different structure, right? And that different structure is now not transparent, right? That's why egg white is called egg white, not egg clear. So yeah, the bonds that hold this structure together they vary in their strength, which is both a good thing, right? Because if they weren't, you wouldn't be able to have the protein change shape, right? And change in shape is pretty key to a lot of protein function. But on the downside, it means that you want to maintain like the ideal conditions for that protein. Otherwise, it's going to change shape, right? And it's not going to work. And so what dictates that shape, obviously, the identity and the order of those amino acids in that polypeptide chain, and what their properties are. And so the properties of an amino acid depend on the R group. Now you're all going to remember it's the R group, right? Because my terrible pirate dad joke. That's see? Teaching excellence at work here, everybody. What can I say? Blow my own trumpet. So these R groups differ, right? And they differ in lots of ways. Right? I'm going to just type up as many of them as I can remember. Good. It's my mad professor skills. Right? So R group properties. And so reason why I'm typing this out is because I don't want you to memorize this table, right? It'll take a lot of time, be very boring, and kind of pointless because you can just look it up on Google. That's what I always do, right? But what I do want you to understand is how these different structures change the property of the amino acid, right? And therefore, the property of the protein, its shape, its function, so on and so forth, right? That's what matters. That's what's interesting, right? And that also kind of ties into the consequences of mutation as well, right? Because if you see, what happens in terms of an amino acid change, you can get some idea as to how that would affect the protein because of what property has changed, right? So R group properties, right? So there's hydrophobic versus hydrophilic. Everybody know what those words mean? No problem if you don't. I just want to make sure I'm not going over your heads. As a cyclist, I am largely hydrophobic. Yes, exactly. Hydrophobic hates water. Which is kind of ironic because as a Brit, you know, I've basically been raised in a land of constant rain. Right. So hydrophobic. Oh, yeah, that works too. Yeah. Philic is like, so if you're an Anglophile, you love British people. And obviously, who wouldn't, right? Because we're adorable. If you're an Anglophobe, or actually, if you're a Francophobe, which most Anglos are, you hate the French. So phobia is hatred of something. Felicity, I think is a word, is a liking of something. Right. So whether or not they're hydrophobic or hydrophilic is very, very important. Right. And so in terms of hydrophobic, all of these ones in yellow up here are hydrophobic, right? So none of these, let's blow them up a little bit, like water. And they don't like water because they have no polar bonds, right? And so essentially, hydro whatever equals ability to form hydrogen bonds. Hydrophilic, oops, equals form H bonds equals bond with water, because that's like what water basically consists of when it's a liquid, a bunch of molecules bound together by hydrogen bonds. Hydrophobic cannot form H bonds, no polar bonds. That's bonds, not bears. Equals repels water. And so that's also what we mean by this polar side chain, right? So which one's this one? Serine. So for example, serine has an OH. That OH, the H, well, the O part of it will be slightly negative. The H part of it will be slightly positive. So you can form polar bonds with other ones of that essentially, right? If you look at the hydrophobic ones, there are no polar bonds here. I said bears, right? All you find are carbons and hydrogens and the odd sulfur group. So put this into a bit of context. Where would you expect to find, given that our cytoplasm is an aqueous environment, right? It's a water-filled environment. Where on a 3D protein, a globular protein like that, would you expect to find hydrophobic amino acids? We're talking about a 3D structure. Yeah, good job, Kaylin. They'll be on the inside, right? They'll be far away from water. And conversely, on the outside, you'd expect to find hydrophilic amino acids, right? And in fact, that's one way in which pressure causes, like barometric pressure, if you go down into the ocean, causes proteins to dissociate, because it actually forces water inside proteins where it shouldn't be, and those proteins fall apart as a result. Okay, so that's one property. What else we got? Get back up there. Sorry. Touchy Excel, PowerPoint. We've also got charge, right? And so these are not small variations in charge, like you can see here, but they are actual differences in charge, right? So you have acidic which are negative, and basic, which are positive, right? Basically, depends on whether they give up a hydrogen, which is what's happened here from an OH group. So this is a carboxyl group, or if they've gained a hydrogen due to the presence of an amine group, right? So another thing is that acidic and basic amino acids, or R groups actually, can form ionic bonds, right, which are much stronger than hydrogen bonds, right? So now you're starting to see how this kind of complexity kind of builds up, basically. What else we got? We've got size. Goes from basically tiny to massive. Right, so up here is glycine, back down you go, which just has a hydrogen for an R group, right? It's about as dull as you can get. And down here, I think this is probably the largest, is arginine, right? Which is this huge long chain of methyl groups and amine groups and all kinds of funkiness. And so if you were to swap out by mutation, right, in the DNA, a codon that encodes glycine, right, so that's hydrophilic and tiny, and instead you get an arginine, which is basic, so it's positively charged and huge, our 3D structure is not going to form properly, right? Because there's going to be something in there that's wrong. It's going to change the properties or how that thing folds together. Right? And you can also form covalent bonds, which are super tough, between two cysteine residues or R groups. And that equals something called a disulfide bridge. Right, so up here you see there's a sulfur group. Well, if you get two cysteines together in the right orientation, pardon me, you can actually get a covalent bond formed between them. Right, and so that locks down the structure, right? It doesn't let it move very much. And so in some places that's good. You want that because maybe you want to hold different bits of protein together, stuff like that. In fact, insulin has a bunch of these, right? So as insulin is sliced and diced in the Golgi body, those disulfide bridges hold the different bits of the insulin protein together. Right, and so essentially like the kind of big picture of what I want you to get is that proteins are super complex, right? They have a very complex and very specific three-dimensional shape. That shape is key to how they work, right? You can get proteins from hemoglobin to something like actinomycin, which is how I'm able to wave my hands around and breathe and do all kinds of stuff as an organism, right? So these are the proteins that slide together as a result of protein contraction, right? And so look at these. Those are totally, completely different from that globular structure that we saw earlier with hemoglobin, right? So they're very different in shape, size, function, what they do, and all of that kind of complexity, right, is encoded by 4 different bits of information, A, U, C's, and G's, in this coding sequence in an mRNA. right? And so what we need to do now is figure out how we go from this information in mRNA to that complex three-dimensional structure. And actually, yeah, I'm just kind of thinking ahead. We've got time. We're going to let's look at a wee video. I'll stick this up. Oh, no, you don't want to watch about machining on a 5-axis machine tool. All right, so this is, it wasn't actually the one I was looking for. One I was looking for was this one. But I'll kind of look through them. This one's nice because it has like someone talking along. But I'll post this up. Actually, let's put this into the chat. I'll add it to the PowerPoint and upload a new version this afternoon with all the other stuff. It's not the central dogma. Sequence hypothesis. Shout at the oh, that's got someone chunning along to it as well. But anyway, essentially, this is after transcription, right? So I'm pretty sure that these green things are introns. And actually, that's kind of, whoa. Easy. That's, whew, slow it down a touch. Come on. There we go. It's kind of funny now that the person sounds like they're a little bit overindulged on the alcohol front. All right, so this is kind of cool because this is what the nuclear membrane looks like from the outside of the nucleus. It has all of these little kind of rosette things are nuclear pores. That's how stuff goes into and out of the nucleus, right? So proteins have to go into the nucleus because they're made in the cytoplasm. mRNA has to get out. So you can actually see it is kind of basically been threaded out of the nucleus through one of those pores. So here's our mRNA, and you can actually see it's been grouped into, so these are the bases, units of 3, those are called codons. Those are our words. So if you think of A, U, C's, and G's as being letters, codons are our words, and they're always in units of 3. So the ribosome binds, so this is the 3' end and cap. The rest of it assembles, and now we get, it processes along the mRNA, adding amino acids to a growing polypeptide chain, right? So these kind of like Santa hats that are coming in are the tRNAs, right? So they come in, let's see. Yeah. It's kind of really weird listening to this in like slow motion. And so, and this is where kind of things start getting complex, right? There's a lot of detail in this. These tRNAs come in, they leave their amino acid behind, which is what these red blobs are. And as the ribosome processes along the mRNA, they leave out the other end, right? So they're charged coming in with an amino acid, and then they leave after they have discharged their amino acid onto the end of this peptide. So there's a lot of stuff going on here. Have a look at these videos. It will give you a good overview of what's going on. They're really, really good. And we'll continue this on Monday. So we've got Monday and Wednesday, we'll finish up all of translation. Okay, cool. All right. Thanks very much, everybody. I hope you have a fabulous weekend. Eat lots of candy, terrorize children, you know, or eat lots of candy and terrorize children by eating their candy, stuff like that. So I'm going to say adios.